So this is this is a large hospital in the northern region of Germany. So we have about 6,000 employers. We have more than 3,000 beds, and we have a huge hematology department, and we have an even more huge transplant service where we take care about all the patients around Hamburg and northern Germany. But for MPNs, we have also patients coming from other parts of Germany or other parts of Europe. So here, but I was asked not to give you a talk about transplant, which is more exciting for me. This is a more dry stuff, you know, and even physicians don't like it very much because it's a lot of bureaucracy and so on. But it's very important for you. So the question was the stages of a clinical trial. So the clinical trial is usually what we do with patients. So this, as Claire nicely mentioned, it has not always to do with drugs. So you can do a clinical trial with psychological intervention, you can do a drug retrospectively, prospectively. So all you do with patients to improve outcome has to do something with the clinical trial. But for the sake of time, and because it's very for you interested, I will focus specifically on drugs, on new drugs, how they should develop, because there is really, and you have seen the nice presentation by Claire, even there are some I would call promising things. Overall, if you compare it to the CLL group just around the corner here, so Ruben nicely mentioned, so we are more in time than then, but regarding new drugs, they are more advanced than we are. You know, they have, because they have more insight into the biology, so we're still missing a lot of biology in our disease, so what's really going on, even if we know there are mutations, we don't know which mutation is first, and we know there are different mutations in different clones. So we still have to learn about the biology, and then we can get more targeting therapy. So nevertheless, so when we have a clinical trial, and this is just a short introduction what we are talking about. So a clinical trial is a systematic investigation in human subjects for evaluating the safety and efficacy of any new drugs. So I focus here on new drugs. And the clinical trials are a set of tests in medical research and drug development that generate safety and efficacy data from healthy interventions in human beings. And actually, the clinical trial are conducted only when there is satisfactory information has been gathered from the quality of a non-clinical safety, and health authority or ethics committee's approval is granted in the country where approval of the drug is sought. So this is very important because many of you maybe know, so this is 40 years, 50 years ago, this was not regulated. And most of you know maybe the story of thalidomide. Thalidomide is also used in myelofibrosis, maybe to improve anemia. It's used in multiple myeloma nowadays. But when the drug was actually used, it was used for insomnia, to get more sleep, because we have all the barbiturates. And then a pregnant woman got the drug because it was more or less for free. There was no clear background, and then we have all this disaster with the miscarriage of the birth. So this was actually the start point, also from the politicians, and to make this procedure more clearly and especially more safety for the patients. So the clinical trial is the mainstay for bringing out new drugs into the market. This is from the pharmaceutical point, and to improve clinical outcome in patients. So this slide I will always repeat because this is a long road from developing new drugs or new ideas into approval. Here it's FDA approval. This is mainly for the US, but we have this in Europe for the EMA approval. So FDA and EMA is here more or less the same. And you can see, if you look to this in the past, so this lasted about more 10, and in some cases also 15 years, when we have an idea and when the drug becomes approved. Okay, so we will start with the so-called preclinical area. In the preclinical area, this is actually pharmacodynamic studies in vivo and animal in vitro preparation. And this is mainly by chance. So you investigated some drugs or some new compounds where you believe this could have some activity in cancer or cancer-related diseases. You tested this drug in animals, or you tested these drugs also in cell lines, human cell lines, and you just want to do some more pharmacological stuff, so absorption, for instance, how is the drug coming to the blood, how is it distributed to the several organs, and how is it eliminated. And this by the renal or by the HEPA, and this all is called pharmacokinetics. So you want to know what happens with the drug in your body, and this is called pharmacokinetics. This is very important. And the other, what you want to see in these so-called preclinical studies, is also toxicity, especially if you use animals. So usually these are mice, but it can be also other animals, where you can see acute, subacute, or chronic toxicity. And then you have the so-called therapeutic index, which you can use for further development. So before you can, this is all without human beings. So if you want to have a new idea, and you have shown in animals preclinical experience that there might be some promising ideas, and you have made all these pharmacokinetics regularly, then you can go. You have to get approval. Yeah, you can go. You can initiate testing in human beings if you can prepare this extensive preclinical laboratory research to the health authority. And this research usually involves many years of experiments in animals and human cells. So in this stage of testing successful, the sponsor can provide this data to the health authority, this is FDA or EMEA, and requesting approval to start testing in human beings. And this is called then IND, or Investigational New Drug Application. And only if the EMEA or FDA is approving this, then you can start with testing this drug or this compound in human beings. So it can happen that at this early stage, it can happen that the health authority is saying, no, this is too toxic, we will not allow it, or they are requesting more data regarding safety, regarding pharmacokinetics. So you can imagine this is a long-lasting, for some painful process. So this is still the phase, so we still go now in an early stage. So this is not in every drug, but in some drugs we have this phase 0. This is so-called microdosing. And this is for some drugs or compounds requested by the health authority. This is very early studies in human, testing pharmacokinetics and pharmacodynamics. So pharmacokinetics is just how the drug behaves in the body. The pharmacodynamics is more how effective is the drug to certain effects. And you started with a very, very low dose. So you do not expect in testing this in volunteers, so in cancer drugs, it's mainly in cancer patients, it's more end-stage cancer patients, because the drug is so low, you don't expect any real benefit for the patient, or in this particular case for the volunteer who is participating in this drug. But sometimes it's required because to avoid toxicity for the next step. So then, when we have this micro stage 0 and we have the animal, then the EMEA or FDA is allowing to start a phase 1 study. And usually you can see a phase 1 study is usually very restricted to only about 15 or 20 volunteers, if it's a new drug for blood pressure, whatever. If it's a drug for cancer or for hematological malignancies, you have to do this in patients who have cancer or have hematological malignancies. You cannot do this in healthy volunteers. But the idea of the phase 1 study is actually to test it in human subjects, but is designed to get safety and tolerability, pharmacokinetics data and pharmacodynamics of the drugs. So 20-25 healthy volunteers, so in cancer patients, in anti-cancer drugs, it's cancer patients. And the duration of such a study is actually quite short, it's 6 to 12 months. And usually this is not blinded and is open labeled, so everybody knows what you get. It's not a randomization. And so the objective is, the primary objective is to test the tolerability and safety and pharmacokinetics, and only secondary pharmacodynamics. And in this phase 1 study, even if the patient participate, though this is more end-stage cancer patients, the benefit for the patient itself is usually very low. So usually the response rate to a new drug tested in a phase 1 study is less than 10% response. It's about 5 to 4% response. But usually in this so-called phase 1 studies, you got an impression where the drug is working most. I give you an example. So when a new compound, there is a compound to the so-called proteasome inhibition. This was a new method how to treat patients. Yeah, they were found in the cells or how they work, and those were tested this in a couple of patients in this phase 1 study. And what they saw in this phase 1 study, actually, there were also 2 patients with multiple myeloma, and they respond very well. And actually, so by chance, they then realized that this drug is working for multiple myeloma and was then developed for multiple myeloma, the next steps, and so on and so on. And now it's approved for multiple myeloma and is a very important drug for those patients who have multiple myeloma. So here you can see some of the development is also by chance. You see something, you expect something different, but at the phase 1 study, is only safety. Safety, it's not efficacy, safety. So the aim is safety, as I mentioned, and what you try to determine in this safety study is also what kind of dose is the proper dose for the further development. And this is actually called the maximum tolerated dose. So we call this MTD. And the MTD is actually developed from the dose-limiting toxicity. This is DLT. So in this particular case, there are different scenarios how you can do this phase 1 study. Usually you have 3 patients in one cohort. You're starting with a very low dose, and you determine your toxicity. So for instance, if there is renal toxicity, hepatotoxicity, this can be graded, grade 1, 2, 3. So usually if you have grade 3 toxicity, then you have to add more 3 patients, and if you have 2 of these grade 3 toxicity, then this is the maximum tolerable dose, and you have to go back to a lower dose. If it's well tolerated, you go to a higher dose. So you will find in this phase 1 study the maximum tolerable dose for the patient. For the next study, which is then the so-called phase 2 study. So, and you can imagine that in this particular case, a lot of drugs or a lot of compounds will not reach the next step. So you have seen this nicely in the slide by Claire, that most of the JAK inhibitors in the development has stopped the development because of toxicity. So they are too toxic, so there was no further development. The next step would be the phase 2 study. So those compounds who have completely reached successful past the phase 1, they got into a phase 2 study. And the phase 2 study is usually a bit longer. The phase 2 study is actually testing the efficacy. So now we have found the dose-limiting toxicity. Now we look for efficacy. And while in the phase 1 study you can use a lot of cancer patients, in the phase 2 study usually particular focus on a specific disease where you believe this could be a good procedure for the next, for the approval. So therefore you have a specific disease or you have a category of disease where you want to see the efficacy. So the efficacy is mainly determined by response. You have response criteria, you have a statistical plan, so how many should get a response and how before you can say this is an effective drug. So usually this is about 30 to 40% should have at least a partial response to determine this is an effective drug. So the second one, what you can test it also is, of course, safety. Safety is always important, but here it's only the secondary objective. So the optimum of the dose finding, the dose efficacy relationship, to look for the therapeutic dose regimen, the duration of the therapy is also an important issue, the frequency of the administration, how you should administrate the drug, and what is the therapeutic window, so when you reach toxicity, when you have low efficacy. So they are coming up more and more new ideas to speed up the process. So we can have, for instance, phase 2A study, but this is quite complicated. It's assessing more dosing requirements, or 2B is designing more study efficacy. But also there are some developments to accelerate this process to get the drug, an effective drug, early into the clinic. There are also some ideas to combine a so-called phase 1-2 study, to combine both phase 1 and phase 2 to accelerate this process, because you can see this is also lasting about 4 years. And if you pass the phase 2 and you have shown that the drug or the compound you are interested in has efficacy in a specific situation, then the next thing to get approval, you have to show that it is either better what have been seen before, or you have to do a so-called phase 3 study. A phase 3 study is usually a large randomized study, where you want to compare your new compound, where you believe it's better, or you can also, depending on the design, sometimes the design is also that it's equivalent, so that is at least as effective as a common treatment, but maybe less toxic. It depends always on the endpoint you have in your study. So in this phase 3 study, this is usually the approval study which you need for the EMEA or FDA, is usually the design is given by the EMEA or by the FDA. So if you have a specific compound where you believe you want to get approval for a specific indication, let's say myelofibrosis or P. vera, then the companies go quite early here in this phase to the health authority and asking, so what do we have to do to get approval? So what is our primary aim? So in the beginning, if you look in the past, it was always overall survival. So survival should be better than before. So in the beginning, when we have, for instance, the ruxolitinib, the primary endpoint was just the reduction of the spleen size by 30%. We were a bit, what is this for an endpoint? So it's not survival, it's what about the spleen, But this has to be discussed with the EMEA, and the EMEA or the FDA is giving the companies in this particular case how they should do the trial to get the approval, so what is really required. So in this phase 3 study, we have then is more a therapeutic confirmative trial. So it's usually a large scale, it's usually a multi-center trial, it's not from one center, multi-center, often also multinational if it's a rare disease. And usually it's also randomized. Randomized means the patients are on a blinded fashion, were dedicated to one of the 2 arms, and neither the physician nor the patient can choose what arm he will get. And if there is, you can imagine that in some instance, because if you have a very new drug, you can have a bias because it's like a placebo effect if the patient feels, oh, this is good. Therefore, in some instance, these trials are blinded. Blinded means that neither the patient nor the physician knows whether the patient got the drug which needs to be confirmed or a placebo or the best available therapy. So this is blinded to avoid any bias in interpretation of the results. So you can see then the target population is much more. So we need hundreds on up to 3,000 patients. So you ask why you need 100 and 3,000. This is depending on the primary endpoint. What do you want to achieve? And the primary endpoint, for instance, if you have a hypothesis that my drug is better than the previous one, and I have a huge benefit, as greater the benefit is, as low a patient you need it. Because if there is a huge difference, you need only a couple of patients to see there's a difference. If the difference is very small, you need more patients. So therefore, it depends on your hypothesis. What do you want to show? And then it can be statistical. It's a statistical method to see whether you need hundreds or maybe you need 3,000 patients to see the benefit you are thinking of and what is your hypothesis with your drug. So this study, depending on the endpoint, takes a long time, up to 5 years, sometimes even longer, sometimes shorter, depending on the endpoint and the disease. And so the phase 3 actually established the efficacy of a drug against an existing therapy in a larger number of patients, method of usage, and to collect also data safety and so on. So just to summarize, so the phase 3, the objectives are to assess the overall and relative therapeutic value of the new drug, efficacy, safety, and special properties. It's also determined the optimal dosage schedule for use in general, and the dose schedule in clinical trials should be as close as possible to its anticipated clinical use. So now, if the phase 3 has been completed, and the phase 3 was successful. Successful means that the hypothesis was verified, that I have reached the primary endpoint. Then usually the EMEA or FDA is approval the drug for this specific indication in this specific setting. So then you have the approval for the drug, and then it depends on the health authority in the different nation and the health politics whether this drug will be reimbursed by the insurance and so on and so on. But you have the approval for a specific drug in either FDA or EMEA. So this is not the same. So it can happen that you have approval for FDA, which is maybe sometimes different in terminology. So for instance, for the Ruxo, the approval in the US is just for intermediate 2 and high-risk patient. Yes, Ruben, is it correct? And in the Europe, it's for symptomatic splenomegaly, which would not exclude actually intermediate 1 patients. So you got the approval, but then most things, then it's finished, but actually it becomes more and more popular to study then these so-called phase 4 studies. And phase 4 studies is actually, we're just coming back to this slide to highlight first the long run to get a drug approved. And you can see on this slide, so there are thousands of drugs being tested in the preclinical setting, in mouse experiments and cell lines, and you can see it becomes less and less. In phase 1, we have even not half of those patients tested preclinical. Phase 2, you can see it goes down even. In phase 3, it's go even down, and at the end, you have only one of these more than 100 drugs tested preclinically who really get at the end the approval. And you can imagine that this procedure not even takes a long time, but it's also very costly. It's quite expensive to do all these drugs, and this might explain why the drug, once it's approved, especially the new drugs, are so expensive. But we can be happy, especially you in the MPN field, we can be happy because if you look back in the past, the industry was not so interested in these rare diseases because the idea was we cannot make a lot of money with these few patients. So, but nowadays, this is good for us who are treating patients with rare disease because the companies are interested to go into these more rare diseases, rare indications. But at the end, it means that the price is always quite high because they want to get this money, what they invest, want to get it back. So coming back to the phase 4 study, the phase 4 study is done after the drug has been marketed. And sometimes this is also called post-marketing surveillance. And there's no fixed duration, no fixed population, but it's usually asked also by the EMEA or FDA. If they approve the drug, they ask the company, so we are quite happy, but we need more data. We need more data about this drug in the real life setting. So that means they're asking for more patients to monitor those patients, but only in this indication, the drug was approved, to get more data about the efficacy, but also about safety and toxicity in the real life world. So the objective of these so-called phase 4 studies is the evaluation in different age group, type of patients, comparative benefit risk assessment. The benefit cost assessment is also important. This is called pharmacoeconomics, and the drug used in the community, and also quality of life assessment. So the phase 3, they should confirm the efficacy and safety profile in a large population during daily practice, but also to detect unknown and rare adverse drug reactions, evaluation of overdosing, identification of new indications, for instance, and dose refining and evaluation of new formulation, dosage, duration of treatment, and so on. So the phase 4 study should harmful effects should be discovered and may result in a drug being no longer sold or restricted to certain use. And this has happened really in the past, that some companies has withdrawn the drug after having seen more toxicity or more rare events occurring in this phase 4 study. So at the end, just to summarize, so it is a long run to get a new compound approved, but at the end it's just for safety of the patients. if you do it too early. So the demand is quite high to push it. So especially in MPNs, we have an unmet clinical need to get new drugs, new efficacy, and new effective drugs. But this takes time and needs to be reconsidered regarding safety. So this is the current. So we have phase 0, I mentioned this microdose, phase 1, defining the toxicity, phase 2, defining efficacy, phase 3, defining clinical outcome, phase 4, seeing long-term outcome and long-term toxicity. So there is the conclusion is there is an unmet clinical need for new effective drugs for treatment for malignant disease, especially for MPNs. But because of this long duration of early clinical testing to approval, they are now getting more so-called new ways discussing with the health authority EMEA and FDA to accelerate this process. And there are several projects like accelerating assessment, the conditional marketing authorization, the so-called compassionate use, and there's also a new project which is currently under discussion, so-called adaptive pathways or adaptive licensing, which always have the intention to make this process faster, to get the drug even more earlier to the patients and to look later on to toxicity. But this is under discussion because it can also at the cost of safety. So you have seen this in the slide by Claire. So for instance, the fedratinib, which was a JAK inhibitor, was always in phase 3 study and was nearly to approval. Then it was realized there are some severe neurotoxicity. There is also some concern about pacritinib, at least some concerns. So you can see safety is important, and safety should be taken into account while developing the new drugs for MPNs. So I thank you for your attention, and I'm open for questions. Thanks.